Hi, folks. So, in this short little section, what I'm going to talk about is some of the basics of the biochemistry, physiology, and really mechanism of action for when we're talking about CBD. So, as you may know, CBD is the kind of non-psychogenic part of cannabis, right? So, THC and cannabis have been, studied for a long time since the 60s, but essentially, for a while, researchers equated basically the psychogenic effects of THC with being active. And CBD didn't have any of those psychogenic effects, so they didn't really study it for some time. And even though you can see in panel B that the actual structure of CBD and THC is very similar, if you're looking at it in a 2D kind of way, if you look on the left-hand side with CBD and THC, the way that this is oriented in space when we're looking at 3D modeling of this makes CBD quite a bit different than THC. So why is that important? Well, that's important because we know that THC binds to endocannabinoid receptors called CB1 and CB2. And there's been lots of different studies based on that. Those receptors can be found all throughout our body, within our brain, as well within the GI tract, and can have many different effects. CBD, the conformation is a little bit different. So when thinking about the binding efficacy of CBD to those receptors, there's still a lot of literature that needs to be done. There's still a lot of research that we need to know about that. So while it can bind, it seems, to CB1 and CB2 receptors, it seems that CBD might, I dare say, act a little more sloppy and bind to a number of different types of receptors. And we'll see that in a minute. So what do we know about CBD that's relevant to inflammation? So in the review paper that I gave you from Burstein in 2015, they talked that they mentioned how it can stimulate arachidonic acid release and favors lipoxygenase pathway. So if you remember, we talked about this early on in the term where inflammatory cells and platelets can release arachidonic acid. This can stimulate the lipoxygenase pathway, which then transfers these products into leukotrienes and ultimately increases vascular permeability. Now, if you're thinking, well, I thought this was supposed to be anti-inflammatory. How is that being anti-inflammatory by increasing vascular permeability? Isn't that one of the key vascular changes that we look at? And it is. So we'll see that there is a lot of different paradoxical kind of effects about CBD going on here. So in this review, I wanted to highlight a few things that they looked at and summarized for us. So they found and looked at a number of different studies, and they really looked at a lot of dose-dependent functions of CBD, which is good, right? So they found that it has a dose-dependent decrease in prostaglandin activity, COX activity, and nitric oxide production after stimulation with an inflammatory agent, okay? This was in rats. So they would give some sort of inflammatory agent. There's a few different ones and a few different models that they did to, induce rat paw inflammation or inflammation within the eye. And then they saw with the different types of doses of CBD, increasing doses, they found that there was a decrease in some of these inflammatory markers. We spent a lot of this course talking about cytokines. And again, if we look to some of the mouse and animal studies, we see that indeed CBD seems to be able to decrease the production of some of these inflammatory cytokines that we know and love. So in a mouse Alzheimer's model, it was able to decrease interleukin-1 beta expression. It can decrease amounts of TNF alpha in rats. When looking in the central nervous system, so the microglial cells. I can't remember if I mentioned this in class or not before, but microglia more or less are like macrophages within your brain. They have a very similar structure and actually have a very similar phenotype as well. And it's been shown that CBD can decrease the release of some of these pro-inflammatory cytokines that are normally released in microglial cells. And then in various different types of disease models, so for example, in different types of arthritis in mice, they can decrease the amount of TNF alpha and IL-1 beta. So you're saying, okay, yeah, this looks pretty good. Here's a few different ways. So we're not talking about mechanism here, but we're looking at correlation, and we're finding that by administering CBD, we are decreasing inflammatory activity in a number of different ways. And what I'm talking about here is looking at with cytokines. Now, when we're thinking about our immune cells, remember, if we want something to have an effect, it has to be able to bind to it somehow, right? If we want to have a downstream effect, we're talking about some sort of cytokine or chemical, it needs to bind to a receptor. Now, the CB1 and CB2 receptors are also expressed on a lot of our immune cells. So you can see here on some of the ones that we know and love, B cells, natural killer cells. You can see neutrophils and monocytes, those being two of the cell types we talked a lot about in this course. Again, monocytes being undifferentiated macrophages that are traveling throughout the bloodstream. CB2 receptor mRNA is also expressed on the above cells, okay? So they were looking at the mRNA, so not necessarily indicating that it has those receptors, but it's producing the mRNA. So again, there could be issues with the production of the CB2 receptor, maybe on the cell surface, but for the most part, it looks like these immune cells have both CB1 and CB2 receptors. And interestingly as well, they think that there might be a potential link between endocannabinoids and suppression of toll-like receptor signaling. So we talked a lot about in this course about toll-like receptors and how really they are so important for stimulating really the production of so many of those cytokines that we know and love. So on this next slide, I'm going to show you a potential mechanism of action. And like I said before, the CBD, can be a little sloppy. It's not just going to bind to CB1 and CB2. And this is from a review paper published in 2018. So potentially what they think is going on is that CBD can bind in the central nervous system to the CB2 receptor, as well as in PPAR gamma. So you might know about PPAR gamma a little bit if you have learned about Alzheimer's disease. But overall, what it is doing is it's decreasing the production of cytokines within our central nervous system. So in the central nervous system, we do have the production of these cytokines. And keep in mind, we've talked about this when we've talked about depression and sleeplessness, all of those things associated with inflammation. And a lot of times those cytokines are going to come from microglia within the brain. CBD can also affect the peripheral system, so within our tissues, macrophages, and B cells, and it can do this by potentially binding to a few of these different receptors. So you can see the CB2 receptor there. The TRPV1 receptor is involved a lot of times in pain transmission, and it can be implicated in creating cytokines that kind of increase nociception. So if you look down the left side of this, again, what you can see is that CBD, through these three receptors, all GPCRs, decreases the amount of COX-2, INOS, TNF-alpha, ultimately decreasing cytokines and decreasing inflammation. Okay, so here's a few different ways of how CBD could potentially work. Now, if you look in here, there's a lot of dashed lines, right? This isn't fleshed out and whole, and there's a lot of questions, obviously, too. Now, I want you to think, as we are going through this evidence, what was the limitation of these studies? We looked at all of them. Do we see any that was looking at humans? No. All of these were done in cells or in animal models. So sometimes human cells, but again, keep in mind that we're looking at a much more simplified system if we're looking at an in vitro culture and trying to figure out mechanisms. Obviously, this is done so that we can decrease the amount of confounding variables. And obviously, for ethical reasons and other practical reasons, we do these types of interventions in animal models as well. So in the next short little video that I'm going to give you guys, we're going to talk about really what is it at the human level that we know about CBD. Is it an anti-inflammatory? Is this something that we can look to in the future, or is this just a whole lot of hype? All right, well, I'll see you guys in part 3.